Good morning. Thank you all for coming. I hope most of you are not too jet lagged coming from Europe. We're going to start our course on parcellation and its relation to function with Simon going to show us some division of the brain based on postmortem features.
So good morning, everybody. Please include me into the people who are jet-lagged. But I will hopefully give you an overview over the next 40 minutes, roughly, on anatomical microstructural parcellation of the brain and how that then relates to function. So in the end, what we want to do is understand how the brain is organized. So that basically comes down to the question, what kind of function does a particular module of the brain have? Now, what determines the functional properties of a particular location in the brain? There are two main constraints. One is the extrinsic properties of that part of the cortex, that is its connectivity to other parts of the brain. And the other factor is the intrinsic microstructure, so to speak, the local infrastructure for information processing. And these together should then determine the functional roles, the functions that a particular part of the brain can actually fulfill. Now, how to do that? There's a very easy scenario how you can integrate structure, function, and connectivity, and that's in non-human animals. Here you can use electrophysiological recordings to characterize the functional properties of a particular location in the brain. You can inject tracers at that very same position and get the anatomical connectivity patterns at the cellular resolution. And last but not least, you can do histological processing and microstructural examination in, again, exactly the same slide. So this is the reason why most of our conceptual knowledge about how brain structure, function, and connectivity relate to each other really derive from non-human primate work, because here we can, in the same individual, at precisely the same location, probe all three main determinants. Now, the problem is these techniques are invasive, and hence we cannot use them in humans. However, over the last 2 decades, evidently functional imaging has made great strides in describing the functional recruitment of particular brain areas in the human brain. Moreover, through techniques like diffusion imaging, resting state, and a whole bunch of other methods, we now get closer and closer to having a fairly detailed picture on the connectivity patterns. But from what I said earlier, we do have 3 aspects that we need to consider. So it's function, it's the extrinsic properties, connectivity, but we should not forget about the intrinsic structure, that is anatomy. And the key reason here really is that without understanding this threefold relationship between internal differentiation, external connections, and the ensuing functions, we will not be able to really understand how the cortex is organized. So in trying to understand how the brain works, we have to consider its structure, and that is anatomy. The problem there is anatomy is multifaceted and multi-hierarchical. So there are many different ways to describe anatomy, and it's organized in a hierarchical fashion. So let me give you a brief primer on neuroanatomy, and I'm apologizing straight away to those people who have a deep anatomical knowledge, but being the first speaker in that talk, in that session, I just tried to pick up also those people who are not really familiar yet with neuroanatomy. So on the broadest level, we have the large-scale lobular anatomy of the cerebral cortex, so the frontal lobe, parietal, and temporal, for example. Now, we all would agree that when describing locations in the human brain, that is way too broad and in fact does not allow any useful allocation, so any useful description of where something is. However, it does have some relevance when you look at model species like the mouse brain, where actually people do refer broadly to the frontal lobe rather than its subdivisions. The next sort of detailed level of anatomical descriptions would be the sulcal and gyral anatomy, so where you label the different fissures and the gyri bounded by them. And this is, in many respects, a useful description in particular when we are talking about regions of the brain that are fairly stable across subjects. So, in fact, the sulci I have labeled here. These are sulci you will find at more or less the same position and with a fairly constant structure to function relationship across basically all subjects. So here it's a very useful description to, for example, say something is located on the pre versus the post-central gyrus. On the other hand, there are regions in the brain, like here in the occipital lobe or the dorsal frontal lobe, where the sulcal pattern becomes much more heterogeneous. So you have a twofold problem here. One is that you may not necessarily recognize that particular sulcus in each particular subject. And on the other hand, also the relationship between sulcal anatomy and the functional organization of the cortex is much more tentative at these regions. Basically covered already. Importantly, brain parts, sulci and gyri, define macroanatomy of the brain. Now, let's have a look at the microanatomy of the cortex. So the basic distinction here is any macroanatomical descriptions, that's things you can see with your naked eye, except, for example, on a 3D rendering of an MRI scan. Microanatomy, however, describes the internal makeup of the cerebral cortex. And there are two fundamental properties that are widely recognized since more than 100 years, and that's cyto and myelo-architecture. Cyto-architecture, and you see a sort of prototypical plot here on the left side, is based on staining cell bodies, nerve cell bodies, and then describing properties of the nerve cell distribution across the cerebral cortex. So here we would have the piled surface, so here that would be the CSF, and you go down the cortex and here into the white matter. And basically when you look across the cerebral cortex, then you have have a layering, so there are prototypically 6 layers that differentiate each other from each other through cell packing density, orientation of cells, type of cells, where you have small granular cells or larger pyramidal cells, and so on. And importantly, and I will go into that on my next slides, the exact makeup, how dense, how large, how arranged the cells are, that varies from one cortical region to another. So we can use that to identify distinct microanatomical entities on the cerebral cortex. On the right side here, you see a schematic drawing reflecting myelo-architecture. So myelo-architecture looks at the presence of myelinated fibers in the brain, in the cerebral cortex. Now we all know that in the white matter, there we obviously have almost exclusively fibers, so it's very myelin dense. But also in the cerebral cortex, we do have myelinated fibers, and here we can look at properties like the overall degree of myelinization in a particular area, or the presence of myelin-dense bands, or the size and density of vertical and horizontal fiber bundles. To give you an example of what I meant with there is a differentiation between different areas, we take here, well, admittedly one of the most striking changes in architectonic pattern, and that's between Brodmann's area 17, so the primary visual cortex, and that's here on the upper left side on each image, and the secondary visual cortex BA18 here on the lower right side. And you can see here in the cell body stain section that in the primary visual cortex you have two dark cell dense bands running around here, and then all of a sudden around that position here that changes. And if you look at the cortex over here, it's missing these dark bands that are full of small granular cells. And likewise, in the myeloarchitecture, you have a very densely myelinated band here, and that then vanishes when you go into the secondary visual cortex. In addition to these classical histological features, cyto and myeloarchitecture, we can also look at a large array of different neurochemical properties, from cytochromoxidase staining to all sorts of neuron proteins, calbindin, and so on, or, for example, as I show here, density of neurotransmitter receptors. So again, we're looking at the visual cortex. Here is the calcarine fissure, so that's the location of the primary visual cortex. And if we move out of that, then we see at some point here the density of the muscarinic M2 receptor all of a sudden drops dramatically, so then we're in a secondary visual cortex. And then at that location, again, a substantial drop, it's V3, and another drop in V4. And now, interestingly, if you look at, for example, the glutamatergic AMPA receptor, we see a different pattern, but a pattern indicating the same transitions. So here the primary visual cortex is a low receptor density, and then when we go to the secondary cortex, the upper cortical layers all of a sudden have a higher AMPA receptor density. If you go to V3, then now also in the lower layers, the receptor density increases and so on. And interestingly, all of these receptor density changes then also align very well with cyto and receptor architectonic changes. So what I'm getting at this is that the cerebral cortex is not homogeneous in its internal, in its microstructural features, that there are a whole array of different intrinsic microstructural properties, neurochemical properties that we could assess. And it seems that largely it is the case that at the border between two different areas, all or at least most of these features changes. Now, it is very likely that many of these features are dependent on each other. So it's not really surprising that when you move from one area to another, all of them change, but it's not also a complete constant. And how these different features relate to each other and how they relate then to what we could call the local infrastructure for information processing. So what does it mean on the functional level that a particular area has a higher cell density in this or that layer? I think we are still quite a bit away from really understanding that. But the key point here is that using a quite large array of different histological techniques, we can identify distinct microstructural modules across the brain. And then the question obviously is, why does all of that matter to me? I'm doing functional imaging anyways. Now, the key thing here is that that microstructural areas really constrain function. So this is a very old example, but a very simple one. So it's a study on tactile versus visual stimulation, and we get this activation here on the left, roughly sensory motor cortex. And from a purely functional perspective, sensory motor cortex is perfectly fine. This is a more than adequate functional description. But if we look a little bit closer, then it does actually matter quite a bit where we are. And this really comes down to the seminal studies by the Parma group in the early 90s that did very detailed investigations in non-human primates. And when you zoom in here into the post-central and into the central region, so here we have the central sulcus, the pre and the post-central gyrus. Yeah? So this is the region around here, central sulcus, pre and post-central gyrus. Then we see that histologically, this region is not homogeneous. But I would hope that everybody, even without a neuroanatomy training, would recognize that this part of the brain here looks quite distinct from that one over here. And in fact, yes, this is the primary motor cortex, primary somatosensory cortex. Now, if we go further along the post-central gyrus, we actually see that, again, there's a change in anatomy around here. So this sort of dark light, dark banding disappears. And there's another change around here, where we again have a sort of dark band down in lamina 6, but it's much less cell dense than over here. Okay, well, the laser pointer is giving me a little bit of a hard time. Okay, so as a schematic drawing, we have the primary motor cortex and then different subdivisions of the primary somatosensory cortex. And down in the central sulcus, you have area 3A, that's mainly related to proprioception. Then we have area 3B, really the primary somatosensory cortex that is containing mainly rapidly adapting sensors, BA1 for movement detection, and then finally BA2 doing more complex integration. Now, importantly, this is based on invasive recordings in non-human primates where people really put in electrodes into these areas and did a lot of different stimulations and really looked at where do neurons respond to what type of stimulation. Key message here, though, is that even in one region that we would consider to be looking from a very sort of broad-scale functional perspective, its primary somatosensory cortex, if we look at somewhat more detail, we do see within that region a microstructural and apparently also a functional differentiation. And this is indeed what I would really present you as sort of the first and major take-home message, is that microstructural areas represent the functional modules of the brain and are hence the most appropriate topographical reference for regionally specific activations. Now, what is regionally specific activations? It should be anything that you measure in functional imaging, because the opposite of a regional specific activation, so to speak, would be whole brain activation, and that's not something you're usually looking for. On the other hand, macroanatomy and stereotaxic locations are not sufficient for the anatomical interpretation of functional imaging results. Now note that I say functional interpretation. Macroanatomy and stereotaxic coordinates are perfectly fine for describing where something is located, but you cannot make any advanced inference about structure-function relationships. So, case in point, you can say that activation is in area 3B, so I really know it's in the primary somatosensory area. On the other hand, saying this activation is on the superior temporal gyrus, it is correct in that you describe where something is, but it doesn't allow you to really nail it down to any particular processing module. That's what I just said. Now, let's look a little bit deeper at anatomical descriptions. And as I said, the big problem here is that they're not mutually exclusive, but rather we have a hierarchy of levels and descriptions. Coming back to the same spot we just had here, the activation in response to touching versus seeing something, we could start on the broadest of all levels in terms of description and say it's located in the forebrain. It's not in the brainstem. Now, that probably won't get you very far when trying to publish activation findings, but it's a valid description. It's in the cortical gray matter, it's in the parietal lobe, it's the posterior wall of the central sulcus. So if you're coming from the sulcal anatomy, here's the central sulcus, it's the posterior wall of that sulcus. Or you can also say it's the anterior wall of the postcentral gyrus. So depending on the perspective you take, you can come up with two different descriptions here. No, it's actually the advancer. That's where do I really have to point that it actually works best? I'll try it. Okay. Finally, we can say it's located in microstructural area 3B. Now, how do we get these labels with a microstructure? Maybe the easiest way is to look it up in the Taylor-Ruck Atlas. Well, there are quite a few problems associated with that particular resource. And for example, it's a very coarse sampling. It's a sparse indication of areal borders, so usually they are not indicated. There's no information on inter-individual variability. It's not an MNI space. I hope that's something that all of you know. The Taylor-Ruck space and MNI space are similar, but by far not identical. But the biggest problem with the Taylor-Ruck Atlas is the last one here. The histological labels on it are completely useless. Now, the reason for that is that Taylor-Ruck and Tenou were neurosurgeons. And the purpose of the atlas is to provide a stereotaxic reference frame. But then when they went to the publisher with that atlas, Springer was basically saying, it's all good, it's probably very useful, but it looks completely boring. Can you actually put something on the slides to make it look a little bit nicer than that? And so they actually took out the old Brodmann map, a schematic two-dimensional drawing, and then said, well, just put that on it here, just roughly. And indeed, the result is visually very appealing. The publisher was happy, the book was published, and it was a big success. But then, 10 years later, 5 to 10 years later, everybody was taking these labels as something real, and they're not. They're really just a visual illustration and just a gimmick, basically. So, since there is really no anatomical basis for Taylor-Ruck labels, and note that I'm not saying that the areas per se are wrong. The areas are based on the histological examinations by Brodmann. But Brodmann looked at the slides, then drew a schematic figure of what he found, and then Taylor-Ruck and Tenou transferred that figure by hand and eye onto their slides, and then people have digitized and interpolated the slices that Taylor-Ruck and Tenou showed to provide things like the Taylor-Ruck demon. So this is not an appropriate reference beyond what you can do yourself when you look at the Brodmann map. So as a strategy or as a way to overcome this and provide a new anatomical reference frame for the cortex, we are working now for many years on what's being called the U-Brain Probabilistic Cytoarchitectonic Atlas. And the idea here is to provide a microstructural atlas of the brain in standard MNI coordinates. So the way that this is done is that we start out with fixed human postmortem brains, and these are then first scanned in the MRI to document the size and the shape of the brain. Then we do histological processing in whole brain sections that are stained for cell bodies. The histological volume is then 3D reconstructed using images of the sections and the MRI taken before sectioning. And now we can do histological examination. So really do microstructural anatomy, both by looking at the part of the brain through the microscope, but also by statistically analyzing cell density patterns across the cortex. Now the identified areas are then transferred to the respective reconstructed sections, and if we then spatially normalize that into the MNI space, we can create probabilistic maps. These are probabilistic maps for histological areas, so in standard reference space, you can get the information at each voxel of the brain, how likely was the particular microstructurally post-mortem histologically defined area present at that particular location of the brain. Now the problem is we get a lot of maps. We get one for each individual area of the brain. And while this is a wealth of information, it does contradict a little bit the sort of convenience you would expect from a brain atlas. An atlas, a brain map, should be a brain with different areas indicated on it. And this is actually something you can derive quite easily from these probabilistic maps as a summary map by computing what's called the maximum probability map. So basically, this map assigns each voxel of the brain to the most likely area at that particular position. So it looks like the traditional brain maps in that you have a continuous non-overlapping parcellation of the brain. But the key difference here is now that this is actually not a schematic figure, but a summary of a population-based, more or less at least 10 subjects, delineation of histological features. And if we look in, for example, here in the primary sensorimotor region, it actually reflects known anatomical relationships very well, like the primary motor cortex with the anterior and posterior part, area 3A, 3B, 1, and 2, basically in the same order as you've seen indicated schematically on the individual slices and from the primates. Now, the only thing that we don't need to do is put that together, this histological map of the brain and our in vivo activations. And there are now tools to do that in at least 3 major software packages. So one that we support ourselves is the OHBM anatomy toolbox, but you also have the same maps implemented in the FSL Atlas tools as well as in AFNI. Now, what can you do with that in order to really get sort of closer towards a multimodal understanding of local brain organization? There are basically 2 fundamental directions. One is that you start out with the imaging and then you allocate your imaging findings to histological areas. And I would like to illustrate that a bit using the amygdala as an example. So if you look at a high resolution image of the human amygdala in vivo, you can see it quite nicely. So it's a 350 millimeter at 4 Tesla. You can see it very nicely, but you don't see a lot of internal subdivisions. Now, if you look at microstructural properties, now this is a receptor architectonic map. We can nicely see a subdifferentiation into different nuclei. And in fact, the same is true when we look at histological sections, so cell-stained sections under the microscope. We again can quite nicely differentiate the different nuclei and nuclei groups. And they're usually grouped into 3 major groups: the superficial, the laterobasal, and the centromedian group. And here you see the probabilistic maps and the maximum probability map. So this map of the amygdala that we have available in standard space and really looks, in terms of all the formats, exactly the same as your neuroimaging data, is actually now based on the microscopic examinations of the cell density patterns in that region and the differentiation of the different nuclei through their histological structure. Now, as an example, I will take out the laterobasal nuclei. That's the group that is shown here in red, in blue. And in animals, it's known that this is basically the, so to speak, input structure into the amygdala. So you have a lot of connections to sensory cortices, neocortical output, and it's really detecting effective and salient stimuli in the environment. So a relevance detection machine. Now, can we see that in fMRI? So this is an fMRI study we did a few years ago, where subjects saw a heard a scream, a yawn, or a laughter, and then first saw a masked face, and then had to judge the emotion present by a later presented face. And when we looked at the emotion presence incongruence effect, we found an activation here in the amygdala, and that was then specific to the laterobasal part. So from that perspective, it at first glance makes sense. What we know from animal studies on the lateral basal amygdala, this is saliency, relevance processing. Now we have an fMRI study using effective stimuli, and we find the lateral basal amygdala. So on the more broad level, that seems to fit quite well, but actually it fits even better when we look into more detail. Because the interesting thing is what we find is that amygdala activation is only present if both modalities, the visual and the auditory, carry effective information. And it's not relevant whether they have the same valence, but it's important that both are effective. In turn, if you have a neutral stimulus, either a yawn or a neutral-looking face in one condition, then you don't get an activation on that spot of the amygdala. And this is exactly what we would have predicted from non-human primates, in that there is a relevance signaling. So it's not a purely bottom-up driven emotion there or not, but there's some higher integration. And in fact, when we followed that up using effective connectivity modeling, we could see that the lateral basal amygdala indeed receives top-down information from sensory cortex. So exactly as we would predict from non-human primates and rodents, we do have this relevance signaling that is resulting from top-down modulation and feed-forward from sensory cortices. Now, where am I getting with that? The key point here is that you could not have made these advanced relationships by basically saying it's the amygdala. I macroanatomically see it's the amygdala, and then you can, but you should not, make all of these inferences and discussions. However, if you can pinpoint things to a particular microstructural module, like in that case the lateral basal amygdala, then you actually open a door to another whole array of literature, which is that in non-human primates and other experimental animals. Because there, it's a long-standing tradition, really, because you can do it, because you can analyze the histology in the same animal, that all findings are related to microstructural areas. So by now also relaying your findings to microstructure in the human, you can actually make that link to non-human animal data that uses the same, so to speak, anatomical reference frame. Now, there is also the reverse, and I think that becomes particularly interesting in the context of brain parcellation tools, as you will hear more about in this morning session, because you can also use these maps as a priori information for function analysis. And again, I would like to use the amygdala as an example. So here you now see a region of interest that is made up by the entire amygdala. And as Danilo will explain a little bit later in one of the following talks, we can do a connectivity-based parcellation based on coactivation maps from that. So in very brief, because I don't want to steal the thunder from Danilo's talk, you basically, for each voxel in that region, compute the whole brain coactivation profile. Yep, so for each voxel in the amygdala, we compute how likely is any other voxel in the brain coactivated with that particular amygdala voxel. And we can then cluster the amygdala voxels into 3 different groups based on this information. And the nice thing is that we get 3 groups, but actually what's really the most relevant information is in this upper left panel here. Here you see the connectivity-based parcellation of the amygdala. Here you see the histology-based parcellation. And as you see, you don't see a lot of differences. Now, I think this is a very important step when looking at in vivo parcellations. This is not to say that you cannot have internal differentiation within a histological area. Think back at what I said at the very beginning. It's the intrinsic properties, the structure, and the extrinsic properties, the connectivity, that together define what a particular part of the brain is doing. So it's in fact not unlikely that within a histological area, that is a unified sort of environment, you have different zones that connect to different other partners. And you could also imagine that two neighboring histological areas share some connectivity information. But the important thing is you should recognize the histological borders also by other modalities. Otherwise, I think we have to be at least skeptical of what we're seeing in vivo, because the histological parcellation can be seen as somewhat of a ground truth. There's no modeling in there, there's no measurement in there. It's really you look through the microscope and see what is going on. And so that can be seen as sort of a ground truth, and I think it's not critical that you do have a one-to-one correspondence in vivo maps. No, you shouldn't have. But at least the major histological borders should also be visible in any in vivo technique. Yeah, skip that a bit in the interest of time. And really just come to my conclusions. So probabilistic cytoactinic maps should be seen as a most valid anatomical reference frame that allows microstructural assignment of in vivo findings. So whatever you find in fMRI, in connectivity, in structural imaging, you can now relate to a particular histological area, and through that also draw comparisons with non-human data that uses almost as a standard histological regions as their allocation. And on the other hand, it will also provide very valid a priori information that you can either use as a starting point, so saying I'm taking one histological area and looking at its internal functional differentiation, or use it as cross-validation because you know that the major areal borders should be recognizable by most of the different techniques. With that, I would like to close. Thank everybody who's been involved in the different project in relation to both brain mapping and the combination with function. And I'm now very happy to take questions. Thanks a lot. I think you were slightly faster.
If you want to get close to the mic.
Can you give us a little update on what happened to these regions that are not classified here?
Yeah, actually this map is a nice one, but it's actually a little bit older one. So there has been quite a lot being done on the frontal cortex. So the maps of the frontal pole are published. There are maps of the subgeneral cingulum. Maps of the anterior cingulum will be published fairly soon. They will be soon available. Now we've done a lot more on the visual cortex, so V3 and ventral and V4, V3 dorsal, V3A on the dorsal part. The posterior fusiform gyrus is mapped, as well as the lateral occipital cortex over here. And no, just by talking, I noticed that although I like this figure visually, it's probably not particularly useful anymore. So take that more as a symbol figure rather than an actual map of what's going on. But I promise I will replace it with a more up-to-date version. So yeah, the visual cortex is much advanced now. Frontal and particular cingulate, there's a lot of things coming. And also in terms of secondary auditory cortices and medial temporal lobe parcellation. So it's actually moving forward. After a few somewhat slower years, it's actually moving forward quite fast again. Yeah.
Can you say a little about how you do this mapping of the individual histology onto the MNI atlas?
Yeah. So it's all volume-based. That's the first major thing. So the mapping of the histological sections onto the MNI reference brain, that was the question. I'm just repeating that for the audience. So you do the mapping in individual sections from the postmortem brain. Now, the first job is to reconstruct the histological volume. Now, we do have an MRI taken prior to sectioning, But the reconstruction of the histological volume, the key factor there is that we do have sections that exactly correspond to the actual slices that we have. Because if we work on the MRI volume, then you basically don't get that one-to-one correspondence to the locations you actually looked at under the microscope. So what we have is the MRI, and we have block face images taken during sectioning. So they are in order, they are subject to the histological processing, shrinkage for example, but they don't have the distortions from the knife yet in it. So basically then the first step is to register the images of the processed slides to the block face images, basically undistorting the cutting and establishing the spatial structure so that they're in order with each other. Then there is a lot of alignment from section to section because although the block faces don't move, the images, if you just register them to the block faces, still wiggle around quite a bit. So there's a lot of linear and nonlinear adaptation to match the sections to the block faces. And then the next step is to match the block face images to the MRI volume to basically get onto the size and the shape that was present before any histological processing was done. Now, if you have done that, you do have the crucial link, because now you have a volume stack where the sections in the volume correspond exactly to the histological sections you looked at under the microscope, but you do have a 3D structure, a 3D volume, that you can now just put into spatial normalization algorithms and get into MNI space. So that is the, I say the crucial part to really get from postmortem histology into in vivo reference spaces.
All right, any other questions? So I have a question. Did you come across two distant areas that can share a similar microstructure in the brain, even if they are like very distant?
It depends a bit on what you call microstructure. So if you look at a single feature, for example, cyto or receptor architecture, cyto or myelo architecture, I would not be surprised. I think we haven't really looked at it systematically, but I would not be surprised if some areas, let's say in the parietal and the frontal lobe, look remarkably similar, though probably not identical. Now, if you extend that into more features, because obviously cyto architecture is just one feature, and there's a limited degree of freedom if you want to follow a general building plan with the 6 layers and so on, then there's just a sort of limited degree of how different you can become with that particular feature. Then it may be that they're similar, but if you look at microstructure a little bit more holistic, so if you have cyto architecture, myelo architecture, receptor information, neurochemical information, then I would feel very confident to say that no two areas look alike. But that is then on the sort of broader multi-feature, multimodal scale. Within every single feature, it's at least possible that they look similar.
Okay, thank you very much.
There was one more.
Another question?
Just briefly, so this figure makes it look like there are boundaries, say, in the parietal temporal lobe, but I always sort of learned that those boundaries are sort of arbitrary based on landmarks. So does the psychoarchitecture actually, as you see it, seem to respect these temporal and parietal boundaries?
Yes and no. So the question is, just for the audience, do microanatomical boundaries respect macroanatomical landmarks? And the answer is a very clear yes and no. So when you come to primary cortices and primary landmarks, then you do have a fairly good correspondence. So the border between the primary somatosensory and primary motor cortex is always at the depths of the central sulcus, with area 3A really being wedged into the fundus, and then 3B and 4 on either side. Same, the primary visual cortex is always in the calcarine fissure, and the border to V2 is just when you move out of that sulcus. Now, the further you go away from these anchoring points of both structural and functional anatomy, and that's actually a very interesting point to it, because it seems that these are really also during development, the anchoring point, that the thalamocortical connections to these primary areas are really sort of the starting point of the rest of the cortical differentiation. So that's why they are so stable also across subjects. If the more you go away from that, the more variable it becomes, and it becomes more variable in terms of inter-individual variance of microstructure, but still you could say you have a main border, but you also have a more inter-individual variability of macrostructure, and the relationship between microstructural borders and macroanatomical landmarks becomes very loose. So that's why I said yes and no. On the primary cortices, on the major landmarks, it's a quite stable relationship. The more you get away from that, the more unstable it becomes.
All right, we're going to move on to the next speaker. Thank you, Simon.